Hey, good morning, everyone. I'll be talking to you this morning about an exciting project which began at the end of January last year. It's called ARCHiOx, which is the analysis and recording of cultural heritage in Oxford. And it's a collaboration between the Bodleian Libraries and the Fiddyment Foundation, which I'll talk about shortly. The project was made possible through the generous funding support of the Helen Hamlin Trust. And in essence, the aim of the project has been to record Bodleian collections material in 3D using prototype recording technology. But why record library material in 3D? Firstly, to make discoveries, we can take an item which is difficult or perhaps impossible to record using conventional photography and create an image which shows just its three-dimensional surface with its color and tone completely removed. This is an image of a copper printing plate, possibly from the early 18th century. And over 300 years, it's become heavily corroded and the design has become difficult to see. But if we remove the tone and color and instead look at a render of the 3D surface of the plate, the engraving is revealed. What's even more exciting is to turn this printing plate over and record the back. Copper plates were valuable and often reused, and we know very little about the front of this particular plate, let alone the reverse. But from the color image, we can see some horizontal lines, but not much more. But from the 3D recording, we can begin to see markings which were difficult or perhaps impossible to see from the conventional recording. Musical notes and words. And if we add some of the original color back in and enhance and mirror the printing plate to make it easier to read, we are now able to see enough details that we can annotate over the markings, which can then be tidied up. And finally, with the image removed, we can now see the full design, a piece of music which was likely to have been inspired by Psalm 9, a brand new discovery made through 3D recording. The data which is being captured isn't just being used to make renders, 2D images which show the surface of an original. The recordings are truly three-dimensional, and this brings us to the second purpose for making 3D recordings, the possibility to create 3D facsimiles. This is another copper printing plate featuring antiquarian Anthony Wood. Again, it's difficult to see. And here's the render of the surface of the same plate with the design revealed. But by loading a single grayscale image, which stores changes in height of the surface of the plate into software, it's possible to see the recording in a different way. Now we can move over the surface of the recording. We can zoom in and explore every engraved line. With this recording, it's possible to create a facsimile. This is a 3D printer in the Factum studios in Madrid, laying down incredibly fine layers of resin and slowly building a replica printing plate. By making a facsimile printing plate, it's possible to use it to produce facsimile prints. Here is the inked facsimile plate. And here's Adam Lowe, founder of the Fiddyment Foundation, pulling it through a replica Goya printing press. This is the resulting print made from a copy of a printing plate, which was engraved 314 years ago. These recordings and the others, which I'll share with you this morning, were made using a new recording system designed and built by Jorge Cano from the Fiddyment Foundation. Based in Madrid, the Fiddyment Foundation are specialists in 3D recording and have worked extensively in museums, galleries, and important heritage sites throughout the world. Here we can see their recent recordings of the Raphael cartoons being made at the Victoria and Albert Museum. Here in Egypt, where the Fiddyment Foundation have been making recordings of the reliefs in the tombs of the Valley of the Kings since 2009. This is the recording system which they've used for the last decade. It's called the Lucida, and it records the surface of originals using a projected laser line and two small cameras. But their new recording system and workflow, which has been used for the first time at the Bodleian, has many advantages. This system works in an entirely different way, recording 3D data through capturing 2D images, and then storing that 3D data within separate 2D images. Named the Selene, it can record originals at over 1 million pixels per square inch and height variation equivalent to less than 1/4 of the width of a human hair. So how does it work? With the camera in a fixed position, the original is photographed 4 times, lit from different directions, top, right, bottom, and left. The image is then analyzed in software in order to determine the surface of the original. From these 4 source images, we can make 3 useful derivatives. Firstly, an image which encodes the direction of the surface of the original at each pixel, using a combination of red, green, and blue to define the angle. This we call the normal map. From this image, we can generate a depth map, which shows the height of the original at each pixel, using a lighter tone for a higher area and a darker tone for a lower area. In other words, we can store the 3D data as a simple 2D grayscale image. By loading the depth map into another application, we can introduce a virtual light source, which we can position at any angle or any height to create a shaded render, which shows only the surface of the original without the tone and color. As the 4 source images are simply 2D color images, these can be combined to make an evenly lit color image of the original 2. This we call the albedo. And this is a big advantage. Previously, using the Lucida scanner, it would have been necessary to capture 3D and color data separately and then align the 2. This slide shows a commonplace model for storing 3D data, a collage of thousands of pieces of an image forming a mesh. And this is a depth map recorded from the same original. It's a far more efficient way of storing 3D data, perhaps 20 times as efficient. And it's because we're storing 3D data in this way that we're able to record at extremely high resolution. But not only are the recordings more efficient, they are also 4 times the resolution achieved previously by the Factum Foundation. Let's take a look at a few more recordings from the Bodleian's collections. This engraved brass plate may simply have been made as a decorative object rather than a printing plate, as the engraved lines are incredibly shallow and faint. Despite this, the plate has clearly been used to make prints, and perhaps because of this, the central design has become very difficult to see. This is a 3D recording showing the surface of the plate, which has revealed the design very effectively. And for small originals, these 3D recordings are relatively easy and quick to make. The image of the render in this example has not been enhanced in Photoshop. This is simply a recording of the original surface. It's not just printing plates that have been recorded for ARCHiOx. Recordings of the prints themselves also reveal details which are almost impossible to see. This Japanese ukiyo-e print was made using wood blocks to apply color. Here you can see the impression as the paper is pushed down onto the wood block from above. If we zoom in, it's also possible to see these textural elements, which were skillfully applied as part of the printing process. Perhaps these can only be fully appreciated through this kind of recording, as they are extremely difficult to see when studying the original and would be almost impossible to record with conventional photography. This beautiful painted page belongs to a 17th century album made in the Mughal Empire. The recording made with the Selene not only shows the individual layers of paint which have been applied to the paper, but also hundreds of pricked holes which can't be seen in the color image. The Sinhalese text from this palm leaf manuscript has been incised using an iron stylus or reed pen before being inked. This is the color recording, a render of the 3D recording, and a composite image combining the two. If we zoom in, we can see not only the incised text, but also the careful pricking around the edge of this deer, which suggests that this design would have been transferred to make copies. This process of copying using pinholes can be seen on the largest original which has so far been captured for the project. Dating to the 14th century, the Gough map is one of the earliest maps to show Great Britain in a geographically recognisable form. This is the render of the 3D surface of the map, captured at over 700,000 pixels per square inch. And in order to capture the map at that resolution, it was necessary to record the map as 85 image tiles, which were then stitched together. This is the highest resolution recording of the map to date, allowing researchers to zoom into specific areas of interest. If we look at the render of the surface, we can see groupings of tiny pinholes. These holes are evidence that the position of geographical features were transferred to the Gough map from an earlier map, precursor map. Studying the holes allows researchers to understand what the precursor map may have looked like and may help to date the map more accurately. Without the recordings, firstly made with the Lucida in 2015, and last year with the higher resolution Selene, this research would be extremely difficult or even impossible. And the new 3D recording that has been used to produce a double-sided, one-to-one, three-dimensional facsimile of the original. So from a medieval map to a medieval manuscript, the illustration at the top of this folio is incomplete, but we can clearly see that the remaining pigment forms the partial outline of a horse. 3D render allows us to see far more of the design. Again, pinholes around the outline, but also these incisions, which have been made using a sharp tool. If we combine these with the markings from the color image, we can start to see the design more clearly. By joining the dots, we can now see the front of the horse, and we can also see the incised markings form a harness. This is connected to two horizontal lines, which along with the remains of two inked wheels, suggest that the design may have included a cart or chariot. Given that the horse has too many legs, and that there is evidence of additional pinholes to the right of the horse's head, perhaps the original design may have featured two horses side by side. The annotation which we've just seen comes from the first folio of this manuscript, dating to the first half of the 9th century, a homily written in Latin with glosses in Old High German. The catalogue describes another annotation in the lower margin of this recto. Hunting scene, barely visible, but clear enough to make a digital annotation. The design was not made with ink, but in the same way as the harness from the previous example. A sharp tool, most likely a dry point stylus, was used to scratch the surface of the parchment. These incised lines are incredibly difficult to see and in most cases impossible to photograph conventionally. And the 3D recording made with the Selene reveals far more of the original annotation. Not just the hunting scene, but also 4 camouflage letters, RODA, recorded for the first time. And we put all of the annotations together from the color image, the illustration from the 3D render, and the newly discovered text. We now have a complete recording of the annotation, perhaps 1200 years after it was made. Other illustrations made using a sharp tool have been found on at least 25 folios from this volume. Another dog in this lower margin. Another one here, just below what appears to be a moon in the right margin. And text too, a capital D. Similar in style to the one from the hunting scene annotation. Likewise, in this annotation, the capital A seems similar in style to the hunting scene annotation. So it could be suggested that they were made by the same individual, perhaps at approximately the same time. Where annotations are difficult to see from a simple shaded render, it's possible to use other kinds of shaders to adjust the position of the virtual light source and to navigate over the surface of the recording in 3D. For each annotation, this has allowed me to be honest and accurate when drawing over them. The impressions recorded from the surface of the next manuscript may not be quite as striking as those from the previous volume, but this volume may allow us to answer some commonly asked questions. Firstly, is it possible to determine when the incised markings were made? In cases where the markings form words or illustrations, language and style may hold the key for researchers, but technology may also play a part, though the initial thought might be that the markings could have been made at any point since the manuscript was produced. In some cases, it could be suggested that they were made before the manuscript was even written. This small region of the lower margin is a good example of how the 3D recording reveals what is almost completely invisible from the color image. An interlaced pattern, very clear on the left, and with evidence of a similar design on the right. Here is the annotation added at the exact position on the color image. And it's clear that these markings are linked to designs used in several decorated initials throughout the volume. This is an example from folio 5 verso. And this one from 38 recto. Is the annotation simply inspired by the decorative elements from the initials? If so, it could be argued that it could have been made at any time. Or is there a direct connection between the annotation and the initial, which suggests that they were created at the same time? It's hard to be sure, but if we superimpose the annotation onto the image of the initial at exactly the same scale, we find that they were very similar in size and form. And so it could be suggested that the impression may have been made by the artist during the stint when the initials were decorated. This upper margin has a far more obvious annotation. The shaded render makes it even clearer. If we look at a greatly magnified oblique view, then we can see that the lower line of the addition has been made through 2 of the letters. Not only has the incised line erased some of the ink as the incision was made, we can also see that the sharp tool used has transferred particles of dried ink to the right. From this, we can determine the direction of the incision, and we know that it must have been made after the text was written. Markings in the upper margin of this folio tell a different story. These lines are fairly clear, but even more prominent in the 3D render. Here, the digital annotations mark the exact position of the markings. But with the annotations removed, you can see that in this example, the lines have not been erased. In fact, there is no evidence whatsoever that the text has been scratched. And it could therefore be suggested that these markings may have been made before the text was added, perhaps during the preparation of the parchment. Up to this point, it's been suggested that the most likely tool that has been used to make the scratch markings would have been a dry point stylus. This would have been very sharp and capable of forming highly detailed text or illustrations. In some cases, these additions are tiny, perhaps just a few centimeters. But given that the lines from the second example may have been made before the manuscript was written, the tool used here may have been different. Perhaps the sort of tool used to rule the page. This would not have been so sharp, broader and more rounded in profile. Taking measurements from the 3D recordings may help to validate this theory. Here I've drawn a line through an incision and a profile has been generated. Both the width and the depth can be examined through this cross-section. This gives us very precise measurements of the tip of the tool used to make the incision. Here, the sharp tool has created a narrow and deep V-shaped profile. While the cross-section of the other example shows a much wider U-shaped profile. This is another example of how a sharp incision has been made into a lower margin. It's another case of the shaded render showing markings which are invisible from the color recording. The digital annotations that show the 2 circular markings, in both cases, the focus of each circle is clearly visible in the render as a relatively deep indentation. This folio has a far more obvious example. And here the circle scratched by the divider is almost complete. possible that the scratch markings in this manuscript may not have been made with a dry point stylus, but instead a divider. In truth, many different tools may have been used to make the markings. Examining profiles through these depressions may be useful for grouping the annotations. This may provide clues as to which may have been made during the same episode, perhaps by the same individual. Here is a V-shaped profile made with the divider or dry point, and a group of annotations made using a similar tool. Measuring the width of the lines is perhaps the best way to separate the annotations. The depth of the lines can also be measured, but there seems to be less consistency between examples. This is due to the minute differences in pressure applied to the tool into the parchment. This is the U-shaped profile made perhaps with a tool used for ruling. Annotations with the U-shaped profile are much wider. The depth of the impressions are generally deeper too, up to 50 microns. So with these new photometric stereo recordings, we can see that it's now possible to conduct detailed analysis into the tools used to make uninked annotations. But do the annotations relate to the text? The dry point annotations recorded from this folio, from the homily which we looked at earlier, are not consistent with the majority of others from this manuscript. Here they've been added between passages of text rather than confined to the margins. Far less obvious and perhaps only recognizable from the 3D render is a small marginal illustration showing two hands tied together with a bow. In order to determine whether or how this annotation might relate to the text, this image was shared with Joe Story, Professor of Early Medieval History at Leicester University. Her interpretation reveals a clear link between the annotation and the text. The text from this section of the homily describes the stoning of Stephen. The connection between inscription and text is most evident from this passage, the translation from which reads, "For many are called, but few are chosen." Passage from chapter 22 of the Gospel of Matthew. And this passage immediately follows verse 13, which includes the text, "Bind him hand and foot." So a clear connection between annotation and text. And there are other hands to be found in the margins of this volume, as well as text and strange non-figurative illustrations. So who might have made these uninked annotations? This 8th century manuscript is a copy of the Acts of the Apostles. It has been a focus of Leicester University PhD candidate Jessica Hodgkinson's research. Linking her research to my work, curator of medieval manuscripts Matthew Holford requested that I record the lower margin of this page. Here is the 3D render, an inscription made using a sharp tool, totally absent from the color recording, and believe me when I say it's impossible to see under conventional lighting. The inscription was enhanced using software and a digital annotation added. So what does it say? What is clear and complete is the name Eadberg preceded by a cross. Which follows is harder to decipher. And this is not the only inscription from this manuscript. Subsequent recordings have revealed the same name, either abbreviated or in full, 15 times. We can even see the name scratched into an initial on the first page. What makes these annotations particularly unusual and important is that they demonstrate an early interaction between a female reader and an early manuscript. An article written by Jessica Hodgkinson will feature in the next AMARC newsletter, which I would encourage you to read to find out more about this discovery. As well as text, there are also many examples of illustrations, mysterious figures. Though they may appear very basic, we should remember that they are incredibly small, and it would not have been possible for the artist to clearly see the marks that they were making. It's been suggested that these figures may have been made by drawing round a finger or thumb. The render here shows another example very clearly. The Eadberg discovery and the figures recorded in this manuscript have received a surprising amount of attention in the press. Perhaps this is because, despite the annotations potentially being made 1300 years ago, we are all able to relate to making these kinds of informal pictures ourselves today. We've mostly focused on recordings made from Western manuscripts, but hundreds of originals from a broad range of the Bodleian's collections have also been captured with the Selene. This painted page from a Persian manuscript, and this 2,500-year-old clay seal sent from a Persian prince to the steward of his estates in Egypt. This impression and 6 others from the collection were used to recreate the lost cylindrical seal which would have been used to make them. Conservators and curators have also explored how 3D renders may assist with condition reporting. Photometric stereo recordings are extremely effective in recording flaking pigment, historic repairs, and insect damage. What's been proven through the recordings made with the Selene is that photometric stereo imaging is every bit as effective at unlocking the mysteries of collections material as established heritage science imaging techniques. Given the range of possibilities which the Selene recordings permit, measurement, virtual relighting, on-screen 3D navigation, and future-proof data suitable for the production of 3D facsimiles, photometric stereo is without doubt a significant new addition to existing methods of cultural heritage imaging. If you'd like to find out more about ARCHiOx, you'll find several articles on the Bodleian blog, The Conveyor, explaining more about the technology and the recordings I've made.
Thank you very much, John. That was absolutely splendid. What a fascinating project and fascinating kit as well. I'm absolutely blown away really by how detailed those renderings are of the surface of the manuscripts, and also how quickly the technology has progressed. I like the comparison between the older, if you can call it old, the older technology and what you're producing now. And I really like that conjunction between the collections, but also the photography, the computer processing, the 3D modeling, the printing of the facsimile, all those technologies coming together, I thought was absolutely brilliant. I'll have a look if there are any questions, but I have one myself. And if people wish to pose questions in the Q&A, please do so. But I'll ask, if I may, to just start things off. I could see from some of those images the striation marks, the incisions made by the knife as the parchment is being scraped. Has your project, or has any work been done on or what might be contributed to our understanding of that process from your project?
Yeah, we've made some initial recordings, some sort of proof of concept recordings to analyze parchment preparation marks, so marks from the scraping of the parchment, and also things like hair follicles as well. Mm-hmm. All of this is just about possible to record. If you can see it in the render, it means it's there, and it means that it's measurable. And certainly hair follicles and things like that are measurable. We can measure the depth of those and the width of those. The scraping marks, it's possible to measure frequency and things like that. So yeah, this might be the focus of a forthcoming project next year if it goes ahead.
Well, I look forward to that. I've got one question in the Q&A from Sarah Griffin. How do you choose which manuscripts to make recordings of? And is this available to books outside the Bodleian? And if so, is there an application process?
So how did we choose? Well, the key is that it has to be relatively flat. This is not, it's not like photogrammetry where we're recording a truly sort of three-dimensional object like a statue, for instance. So we're really just capturing the surface, just the first 3 or 4 millimeters. So if it's relatively flat, like library material is, then we can record it. But we started off with printing plates because obviously most of them are very flat, but they do have texture and they're very, very difficult to record conventionally. And then it kind of, I was open to suggestions from curators, conservators, and researchers, and that's kind of led a path to where we are at the moment. So, for instance, the scratched glosses were not actually in the original proposal. It just so happened that we were getting great results, and so we continued making recordings from those. And then to answer the second question, yes, we can record other institutions' originals, and we've just started that process. So it will be added as a product to imaging services products and will be available for everyone.
Okay, I've got a couple more questions. Just to follow up on that, something from Claire Burridge. How expensive is this technique? And is it possible to roll it out more widely?
The machine itself is expensive, but part of the price includes a very high spec computer and software which you would require to make the recordings and to do the processing. So sales of the Selene are probably going to happen in the next few months, and that is being organized by the Factum Foundation. So although we've participated in making the recordings, we're not really responsible for selling the machine itself. That would be the Factum Foundation, but it will be available. It will be expensive, but so are any sort of imaging, high quality imaging systems. So yeah.
Great. A few more questions. One from Gwen Riley-Jones at Manchester. What kind of files do you use to preserve the digital objects, and do you have metadata schemas developed for this capture technique? Okay,
so they are just two-dimensional TIFF images, both the 3D recordings, like I was describing, we're saving 3D data as just 2D images. That's the beauty of this system. It's not some sort of intangible, weird file format. It is just an image. So we'll have a separate image for the color, the albedo. We'll have a separate image for the depth map and the normal map. And that will be surfaced, we hope, on Digital Bodleian fairly soon. And actually, colleagues at the Bodleian have been busy making virtual relighting tools, which are going to be embedded in the IIIF manifest. So that should be very interesting. In terms of metadata, it's kind of early days, really, because raster maps, I don't think, have really been made for cultural heritage purposes before. So we'll see how we go on that one.
Great. Thanks very much, John. I think it might be time to move on to the next presentation. There are several other questions in the chat, but we should probably move on. So I apologise to anybody whose question hasn't been answered, but John, you'll be happy to be contacted.
Please do.
Directly. So, okay. So our next presentation is from Sarah Fiddyment and Matthew Teasdale. They are postdoctoral research associates at the MacDonald Institute of Archaeology in Cambridge, and they both work on the project Beasts to Craft, biocodicology as a new approach to the study of parchment manuscripts. Sarah and Matthew, over to you.
We're not able to share our screen currently.
Could our kind of technical wizards at the Bodleian let you? Yeah. Okay, you should be able to do it now, Matthew.
Great. Well, thank you, James, for inviting us to give this talk. So we're going to talk to you about the Beasts to Craft project, which focuses on the methodology of biocodicology. So many of you may already know about it, but for those who haven't come across it, we've coined the term biocodicology to encompass the study of biomolecular information that forms part of the biological history of a manuscript. So, as we've just mentioned, we have been working on the European funded Vista Craft project, which is ending very soon. But it has basically been looking at parchment as a medium through various different perspectives, from conservation, scientific techniques, just to get us a better understanding of parchment as a material and all the biological information you can gain from it. So if you're interested in looking a little bit more about our work, everything's on our website. So from a biocodicological aspect, there are two biomolecules that we mainly work on. So we're going to be talking about today proteins, which is what I specialize in, and then Matthew will talk about DNA, which is his field. So when you approach a biocodicological study, the very first thing you have to think about is actually how you're going to sample the object. So we use a non-invasive technique using triboroelectric extraction, but basically using PVC erasers. So this technique was developed in conjunction with the conservators at the Borthwick Archive in the University of York. And it's an incredibly easy process. You literally take an individual piece of PVC eraser, you gently wipe it on the surface of the parchment, and then all the little crumbs that are generated are the ones that you usually sort of blow away. Those are the ones that you then collect and you put into a sterile tube, and that is the sampling technique done. That tube is then sent to us at our labs, and from there we're able to extract both proteins and DNA. It's been readily adopted by the conservation community because it is a methodology that has been used previously to clean parchments. It's very simple to perform. It doesn't require any special instrumentation. And once you've done the sampling, the sample is incredibly stable. You don't need to do anything to it. So you can literally just put it in a box in a drawer somewhere, and then when you're ready to analyze it, you send it to us. So it's a very convenient methodology for people to be able to collect these samples while they're often doing other forms of conservation work. So what kind of information can we get by looking at these biomolecules? We're going to focus on three main elements. So the first and most obvious one is to do with species selection, to do with the animals used to make the parchment. There's also another aspect that we're exploring on the actual use of manuscripts and what are the proteins you can find on the surface of these documents. And finally, what can it tell us about the environment, both of where the manuscripts were produced, but also their subsequent history, where they've been stored, and their life and use. So the first example we did of the species analysis, shall we say, using this technique was looking at 13th century pocket Bibles. So these Bibles are famous because of their incredibly fine parchment leaves, almost like tissue paper, and there had been various theories as to how they were produced to this fine quality. Was it use of specific animals such as rabbit or squirrel? Also the use of, for example, stillborn calves. So we sampled about 80 of these pocket Bibles and also contemporary documents throughout Europe that weren't pocket Bibles. And what we were able to discover using really only species identification was that actually, no, there are no exotic animals. They're not using rabbits. They're not using squirrels. They are using sheep, calf and goat, the main staples of parchment production. And it's very much tied into where it's being produced. So they are using what is accessible to them locally. So there is a sort of a more of a predominance of the use of calf in France, which is mirrored in other documents. Goat appears more in Italy, again, also mirrored by other contemporary documents. England is a slightly more complex story because you do find evidence for all three. But basically what we could say using this very simple method was this is all down to a production technique. This is a craft specialization that they developed where they could use regular skins but work it down to create this incredibly fine material. For the question of were they using stillborn calves or not, we did a subsequent experiment which For this case, we did have to use destructive sampling, so we were using modern samples. We used a modern stillborn calf given to us by Jessie May in Pergamena, and also a piece of 18th century parchment that had been donated for destructive analysis. And basically, by doing a more complex experiment, we were actually able to measure the different proteins present in this stillborn calf versus a non-stillborn calf. And what we were able to show is that we could actually distinguish between the two. So particularly things like fetal hemoglobin and a type of collagen, that collagen 3, which decreases with the age of the animal, we were able to see an explicit difference. So there is the potential to actually identify stillborn animals. However, at this point, we can't do it non-destructively. Another useful example of where species identification has become quite handy is with exotic animals. So often things may be catalogued under the assumption that they are animals of slightly more exotic origin than they really are. So we were given an example here of an 18th century Ethiopian Bible case from the National Leather Collection, and they had it documented to be possibly made of gazelle skin. However, when we did our analysis, it came back as sheep. So this has often actually been the case for us, where we are given a particular sample that is thought to be of one animal, but in general, calf, sheep, and goat definitely make up the largest proportion of animals used. So another example of how species is important, but in slightly different ways. In this particular case, we were given the opportunity to analyse this 12th century gloss Gospel of St. Luke, but we were able to do the entirety of the document. So we were able to sample every bifolio and also the bindings, as it was in its original bindings. And what we found was in this one document, in this one book, we were able to find 5 different species of animal composing the object. So we were able to determine that the binding was actually made of roe deer skin, and the strap that held it together was actually red deer or fallow deer. We can't differentiate between the two, but it was certainly a different deer to the actual cover. In the inside, the text block is all parchment, and what we found was a combination of calf, sheep, and goat. But there seemed to be a real structure to the way that these were put together. So you can see that the quires, the first half of the book, are quite determined that they interlace calf and sheep. They have one the wrong way round, presumably it was folded incorrectly. There comes to a point in sort of quire 11 where they're using all calf. Then they start to introduce a little bit more sheep. Then we have 2 bifolios made of goats. It's actually been reconstructed by Jerzy Winicek, our collaborator, and he could see that it's the back half of a goat, and it's actually quite poor quality goat, but that appears there. Then we go back to the original structure, and then from then on, we only have sheep parchment for the rest of the book. Now, what's really interesting is on quire 15, where we have this last regular format before the sheep, the paleographers who have looked at this book see that there's a change in the scribal hand. So the main scribe stops writing, and another scribe takes over, which they suggest is a worse scribe. His writing is not as good as not as proficient as the first scribe. And it's interesting that at that point, he is given only sheep parchment to write on. Now, whether that was an accidental choice because they didn't have any more calf, or it was deliberate because parchment is cheaper and not considered of highest quality or as higher quality as calf, we don't know. But it is interesting that that change would happen exactly there. So just from looking at this one object, we started to get a real insight about how this book was put together, what animals are being used, and how they think about the actual process of bringing a book object together. We've also been able to look at species for other reasons in the case of treatment of parchment, for example. So we've been asked on a couple of occasions to actually look at parchment folios that have been dyed or are coloured purple or bluish. And in the cases that we've looked at, they've all turned out to be sheepskin, which is interesting in itself. But We've also linked this to a modern study that has looked at the lipid contents of different parchment made from different animals. And what was interesting is that the people who made this study only looked at the lipid qualities, but they didn't look at what animals they procured their parchment for. But when you look at the distribution of those parchments, which you can see in the graph down here, the parchments that had the highest quantity of lipids or fats were the sheep, and then the calf and goat much lower. So it's interesting to note that the pages that are being dyed this blue-purple color are the sheep ones, which also have a much higher lipid content. So could this actually be a deliberate choice because they have a better uptake, for example, of this dye? Or is it a regional thing? And also because of the area of production, sheep is dominant in that area too. But it is something to bear in mind. So apart from the species, as I said before, we are now starting to expand to look at what other protein evidence you can find on documents that can give you more of an insight into how these books were used. And this is a study that we published in collaboration with the Wellcome, looking at an English medieval birthing girdle. So this birthing girdle is made up of 4 pieces of parchment glued together. It's quite long, over 2 meters long. And it's supposed to be used by pregnant women as a sort of talisman or amulet to basically protect them during pregnancy and birth. So we were able to do some analysis of some of the stained areas on this document. So initially, apart from identifying the animal species of parchment, which turned out all to be sheep, which again made a lot of sense because sheep is quite pliable parchment, so easy to wrap around. We were also able to detect a whole host of proteins that were present on the surface of this document. So we were able to find human ones and non-human ones. In the case of non-human, we were able to detect the presence of things like honey, sheep or goat's milk, eggs, both the egg white and the egg yolk, and various leguminous plants. We were also able to detect mouse urine, but we're pretty confident that that's probably a subsequent environmental incursion. And all of these non-human proteins all are documented to be part of recipes or treatments for pregnant and birthing women. So they are all things that would have been used in contemporary situations. In addition to that, we also found a whole suite of human proteins on the surface of this girdle. And when we matched it to different types of human tissues, what we found is the one that they matched most closely to was cervicovaginal fluid. So it seemed to indicate that the girdle was actually being worn by pregnant women, probably in late pregnancy, and actually wrapped around them as a supportive girdle. So it was really a wonderful discovery to see that there is all this biological information on the surface of documents that you apparently wouldn't see, but is telling us so much more about the history and use of these objects. Now I'll hand over to Matthew for the DNA.
Yeah, so my background is in animal genetics. So I'm an animal geneticist by training. So I came to this project in a sort of tangential way, interested in how we can use the parchment record as a source of animal genetic information. So I really like these 2 quotes, one from the head of our project, but also one from a recent book, describing or making you think again at the archive. So we have, this is particularly when we went to the Borthwick, there's shelves and shelves of parchment documents, which are shelves and shelves of animal skins. And could we use those this biological record to understand how animals have been managed in the past and something about their genetics, and then could we devise other methods that might be useful for the study of the manuscripts themselves? So the first thing we had to do was try and see, could we use Sarah's eraser method to generate DNA data as well as proteomic data? And this is just a side-by-side comparison of documents that we were able to use for destructive analysis compared with the amount of data. So the axis on the side is the amount of DNA we can retrieve from the documents, with larger numbers being better. And you can see that the side-by-side comparison of the eraser method versus sampling a bit of the parchment. In some cases, the eraser method is even better than cut pieces. So we were very happy that the data we were generating from the eraser crumb sampling is useful to do a genetic analysis. So that allowed us then to increase the amount of parchment we could do these DNA analyses with. And the first thing we were thinking about was, could you use the genetic tools that we have at our disposal to see, can you place documents within a genetic map? So the two maps on the left-hand side of this slide, the first one is a collection of modern sheep populations that have a particular geographic origin, so they're linked to a particular region, and that's their physical map. So that's where in the current globe they were sampled from, and at the bottom is their genetic map. So if you take genetic markers that we're interested in looking at, and then you make a 2D representation of that, you can see that the genetic map has a distinct pattern to it. So we have Leicester animals at the bottom of the PC3, and then we have the Scottish Isles at the top. So we were hoping, can we then use that genetic map to place a document where maybe its provenance was unknown back onto the genetic map on the bottom, which would then give us an insight into where that animal came from in a particular time. So the first study we did was from two documents from York. On the right is a 17th century document, and on the left is an 18th century document. And you can see warmer colours in this map are more closely related to the sheep that produced the parchment. So in the 17th century document, we're getting a resolution to sort of the north of Scotland and the Northern Isles. And this represents a sort of a more a different genetic animal, a different type of sheep than in the later documents where we're seeing a resolution towards Leicester. And there's well-known movements of animal genetics from the Midlands into the north of England at that time. So are the parchment samples, are they not just a record of the textual information that produced the document, but also a record of the genetic information of the animals that we use to make the document? And I think it shows it quite clearly that you can see, if you look at documents through time, you are seeing a genetic change, and this suggests that these documents are a somewhat unintentional record of animal genetics to at least from the 1300s to the 1800s. And this is a nice study that sort of uses the method in a different way. So this is, we had nothing to do with this study. This was done in New Zealand. And it's a really nice analysis, I think, that shows the power of, if you have an interesting question that you want to answer, the genetic methods can help you answer it. So this is from the founding documents of New Zealand. And they had there's a bit of off-cut parchment that's travelled with the document, and it's thought to have been from the original document, but then cut off. And they did a genetic analysis that happily confirmed that, yes, this off-cut that has been preserved is the same animal as the one that was used to make documents. This is a nice way of using genetic studies to sort of piece these two objects back together, so we can confirm that they were once part of the same skin. And then the second sort of feature of our genetic analysis we do on parchment looks at the microbiome on the surface. So again, this is kind of an unintentional archive on the surface of the documents that have been preserved. So as we know, parchment is skin. The skin, human skin, animal skin, have their own collection of microorganisms that live on the surface. It's their home, it's their ecosystem, it's where they like to live. And this two slides just give you a sort of an overview of how different communities of microorganisms live on different parts of the human body. But for our sort of purposes, what we're interested in seeing is, could we, if we look at the DNA from we don't just get the animal DNA, we also get what's on the surface of the document as well when we're sampling with the eraser sample. Could we tie that back to particular communities, and could we say, yes, we are seeing skin or some other human microbiota on the surface of the document? So to do this, we got the sort of unique opportunity to sample the York Gospels. This was a particular interest to us because it's a living used document, so it's constantly in use, it's constantly being handled. It's been handled for over 1,000 years. So has this handling left the signal on the document? So it was a really interesting example that we could use to try and test our microbiome method. And what we did was we placed the parchment samples onto a modern day map. So the circles you can see on the top diagram in the sort of faded out colors are all from modern humans. Anonymized data from the Human Microbiome Project that tries to track different different communities of organisms in different parts of the human body. So this data is freely available and other researchers can then use it to study their samples, which is what we did. So we placed our parchment samples on top of this map to see what does the microbiome of parchment most closely resemble. And in this analysis, we found that the microbiome most closely resembles human skin, and also you can see human nose. When this is probably, as everyone knows now, if they've tried to do COVID sampling of their nose, you don't really stick the swab too far up your nose. So it's probably a big overlap between the nose microbiome and the skin microbiome of that region. So we're happy that that's not a false positive. We just think that it's the skin and nose is probably quite similar. So the interesting sort of result of this is that it's sort of a self, you would expect it probably going in, that the microbiome of the surface of the document resembles human skin. So it was nice to be able to show that, but it's also interesting to think about where does this come from. So it's one of our open research questions at the minute, is this all these communities set down when the document's made? So could it be a marker of document production, or is it handling? So does human handling, does every reader, everyone who handles a document leave some of their skin microbiome on the surface of the document, and that's what we're picking up. It also has important preservation and conservation aspects. So if you look at the bottom diagram in B, the one that's most heavily in red is Saccharopolyspora. So that's a species, a genus, sorry, of microorganism that can eat collagen. So in the right conditions, if the document gets damp or other sorts of insults, I suppose, happen to the parchment, these microorganisms can digest collagen, so they could damage the document. So by looking at the microorganisms that are on the surface of the parchment, we could say something about the conservation state of the document. Is it something to be worried about? Because the ARCHiOx doesn't seem to have evidence of purple spotting, but in the future, and other parchments, that maybe this method could sort of highlight possible conservation problems that might happen in the future. So by doing the comparison, we're really looking at, one, the use, the life history of the document, but also maybe able to inform something about its conservation. And this just a sort of an end point, which sort of has resonance for some of the talks that are coming as well. How one archive can be an unintentional archive of something else. So there's this really nice study looking at the UK seed bank. So the seeds were collected for their use as seeds, and then to hopefully be able to either study their genetics or use them to regrow species that might be lost. But in collecting the seeds, they were also collecting all the fungi that lived in the seeds. So people who were interested in looking at the diversity of fungi for particular animals, in this case bananas, were able to use the seeds that were collected from one purpose to understand a different scientific question altogether. And in some cases, and this sort of has resonance for when we think about when we study, well, our study of documents, where the documents were initially collected for their textual reasons, but our analysis is uncovering other layers of information that were also unintentionally kept or have now been kept. So the archive is not just a textual archive, it is now a biological archive. And I think it's nice to think of it that way as well. It has this double life, I suppose. Yeah, and it's sort of it's finding resonance in other studies that are looking at other archives as well, how an archive kept for one thing might actually be very useful for something else. And then that's just a thought to leave you with, I suppose. And then our acknowledgements, yeah, is to say thank you to everyone who has participated and helped the Beastercraft project and future projects and previous projects. We think we're in somewhere in the region of 663 libraries and 42 archives. A big thank you to Matthew Collins, who's the PI of the project, and to the Richard Lansbury Foundation and the ERC for their funding. And thank you for your attention.
Thank you very much, Matthew, and thank you, Sarah. That was really interesting. What a great presentation, and an absolutely fascinating conjunction between sort of the materials and their contents and their use with the birthing girdle. I can see that sort of analysis could have really interesting potential for the study of medical manuscripts and medical recipe compilations and that sort of thing. So, okay, I'll turn this over to the floor and see if anybody has any questions that they'd like to pose. While we're waiting, I will just explain to everybody that at 10 past we'll have a break until 12:30. You don't need to leave the session, but you can just tune in when we're back. We'll all turn our cameras and microphones off and have a break, and then we'll come back at 12:30. So Isle Poleg has just raised a point that the York Gospel has also been used for oath taking, which may have impacted its microbiome. I suppose, yes, I suppose I'm inferring from that he's implying that people speaking over it is going to have an effect, that residue is landing on it as a consequence. And this is something I imagine Kate Rudy being particularly interested in, that sort of interaction with the surface of the page. And yeah. Oh, Keira, go ahead.
I thought I could just jump in and ask a question. Sarah, it's a question for you actually around the plant proteins. Is there any way to kind of get an idea from your data as to what part of the plant that comes from, or if it's a plant product, or can you go any deeper with the data from that?
Yes, so the useful thing about proteomics, which we haven't talked about, so with proteomics, you get the actual tissue specificity. So with the DNA, you just get the species, but we can tell exactly where that protein comes from. So often in our cases, we're finding globular seed proteins because they survive better. One of the things that we're interested in looking at actually is the protocol that we use at the moment for extraction is not particularly set up for plant proteins, so there's probably more that we're not getting. So there are different sort of extraction techniques and different enzymes you can use to actually try and pull out more of this plant material. But yeah, you could definitely say where these proteins are coming from, what tissue, what part of the plant. At the moment, usually more seeds.
So you could tell something if something is flour versus wheat, you could tell if it was a kind of plant product versus a
well, it depends. It depends if in the flour, if the same protein appears in the flour and in the wheat, if you see what I mean. Like, for example, we can tell egg white from egg yolk, for example, but if there's a protein that appears in both of those, then you wouldn't be able to. So it depends on the specificity of how much, So you could tell different parts, but yeah.
That makes sense. Fascinating. Okay, great. Thank you.
Okay, audience, are there any more questions from you? There's nothing. Something from Claire Burridge. So on the microbiome, do you have any thoughts about the variation in relative abundance of different bacteria throughout a manuscript? So she's thinking about the image on the third or fourth to last slide, if that helps.
So yeah, we sort of, it's an open question. Is it that if you handle the document more, are you leaving more? Is it a heavily handled page have more microbiome than other pages? Or does it sort of remain consistent? I would say we tend to target two different things. I suppose when we're targeting the microbiome, we want like the heavily handled pages. So we've never done a systematic look to see how it varies on a page, which would be an interesting thing to look at. We've really only done it, we've been sampling, I suppose, because we wanted to find the microbiome. We haven't sort of done a sort of a wider study. We're also looking at paper versus parchment as well to see if different microorganisms can live on paper versus parchment. Would that tell you something about the conservation history? So yeah, it's a great question, and it's definitely something we're still sort of wrestling with, I suppose. Is we can see it, but we can't. We're still wanting to know how it comes to be there, and can it tell you more interesting things than just there are all these bugs on the surface of the parchment.
In the Philobiblon, Richard de Berry complains about people handling manuscripts with their sort of filthy leather gloves and their dirty fingernails and their greasy hands. But I suppose little did he know that actually that is now furnishing us with really useful information.
Yeah, sounds great.
So Anne Ramon has asked, can your research find DNA for ancestry type DNA matching?
So my interest is we're primarily focused on the animal DNA and not on anything else. So we don't look for sort of In microbiome studies, you tend to remove any DNA that's not microorganisms, and for the animal genetic studies, we just look at the animals. So it's not something we're interested in doing, and it's not something we have done. Yeah, we're really interested in using the animal genetic DNA to say something about. My main interest, I suppose, is it's using this extensive record to track animal genetics through time. So it's thinking about particularly regional archives, so like the Borthwick Archive and the say the Norfolk Record Office as well. That's an incredible collection of animal genetic information. And if you think about how much time and effort goes into collecting for the seed banks, for the big one, the World Seed Bank, but also the one in Kew, that collection of useful genetic information is already in the archives. So our interest is in how we can sort of develop tools to understand the genetic information that's there for the animals. So our focus is more on that. We haven't, we don't tend to look at the other genetic information.
We've had a request from Jenny. Some very helpful publications have flashed by. Can we have a bibliography of these, and including the papers that come after the break? I think perhaps what we'll do, Jenny, is we can circulate the recording of the webinar to participants, and we could include bibliographies from the presenters at the same time. So yes, let's do that. Okay, so it's 12:10. So I think we'll break there. So as I say, you don't need to log out of the meeting and log back in again. You can just leave things running, but the participants, we will turn our cameras off and put ourselves on mute while we go and have a cup of tea or something, and then we'll come back at 12:30 for Keira and Eileen's paper. Okay. Okay, welcome back, everybody. And our third and fourth, because they're doing it jointly, speakers today are Eileen Tisdall, lecturer in environmental geography at the University of Stirling, and Keira McKee, book conservator at the University of Glasgow, who's going to start things off. Over to you, Keira.
Okay, if I just remember to unmute myself, might be a good start. Okay, let me just try and share my screen. Okay, how's that? Is that
perfect? Yes.
Okay, all right. So in our talk today, Eileen and I are going to be looking at the intersection between two distinct projects. So firstly, is a conservation project based at the University of Glasgow, which is an NMCT funded project. And that was managed by myself along with my mentor on the project, John Mumford. And then secondly, there's a Leverhulme project managed by Professor Kate Rudy at the University of St Andrews, which she's been working on with Eileen Tisdall at the University of Stirling. So just for context, we will outline each of those projects briefly. Each of them could be its own presentation, so it'll be quite brief. And then we'll look at the different approaches that we've taken to gathering gutter debris for the purposes of looking for pollen, why we're doing that, and some sort of initial findings, lessons learned from what really is a first attempt at this process. It's just our first toes in the water. So when Cortes first arrived in 1519, the Aztec Empire had been developing for around 150 years. And the Mexican plateau was largely populated with its inhabitants, with a notable exception being the state of Tlaxcala. So this state had managed to defend quite a sizable territory, and when the Spanish arrived, the Tlaxcalans allied themselves with the Spanish and became a key ally of the Spanish in the war of conquest against the Aztecs and other states. So the Historia de Tlaxcala contains a detailed narrative of the state of Tlaxcala from the start of the Spanish conquest, and it can be thought of in three parts. So firstly, there's the narrative section, which is referred to either as the description or the historical chronicle. I'll probably refer to it as the historical Chronicle today, if I can. And this is an extended response to a survey that was sent out by King Philip of Spain, King Philip II of Spain. And it contains information on demographics, terrain, and lots of different areas. It's thought that this is a fair copy of an earlier text that was written out in Spain, which I'll come to in a moment. And then this is accompanied by an illustrated chronicle, which recounts the events and exploits of collapsed Pollen in 156 drawings. You can see one of those here on the right, largely focused on scenes of battle and conquest. This section, there's some debate over its provenance, whether it was produced. It's mostly believed that it was produced in Mexico by indigenous painter scribes, quite a number of them. But there's also some debate about that amongst art historians, whether perhaps it may have been produced later in Spain along with the other portion of the manuscript. These two sections are accompanied by two inserted wheel calendars, one of which you saw on the opening slide. So there are some questions we have about the provenance and where each section of the manuscript may have been produced. In terms of provenance, what we think we know so far about the manuscript is that it's generally believed that the three parts of the manuscript were brought together in Madrid in 1580s when the author of the historical chronicle, Diego Muñoz Camargo, accompanied a delegation to Spain to kind of campaign for privileges that they felt they were owed by the Spanish. And that it was bound and presented as a diplomatic gift during that visit. We know that it remained in the Biblioteca Real at El Escorial until at least the early 17th century. We know it was there in 1629, but after that it gets a bit vague. There's a manuscript note in what's believed to be a 17th century hand, says de la Libreria de Hortensio, so from the bookshop of Hortensio, that's not been explained to date. Then we know that it's purchased by Dr. William Hunter for his museum in Glasgow in 1768, but where he acquired it from or when it left El Escorial is not known. And then it becomes part of the University of Glasgow Special Collections in 1807 with the Hunterian bequest. So it had been unavailable for research for a number of years owing to condition issues. So you can see on the previous slide, if I just pop back, you can see that the parchment cover is really distorted and shrunken. It's not protecting the manuscript or the foredge anymore. The sewing had broken down at the front and back of the manuscript, and we also had numerous instances of advanced ink corrosion. So a treatment plan was agreed with the librarian that would see the central portion of the sewing stabilized, and then the parchment cover kept separately, and that the binding would be, or the manuscript would be placed into a new binding that would offer suitable protection. Once we remove that cover, we get some more provenance questions. So it had always been believed that that parchment cover was the original binding. That's how it was listed in the catalogue. That's what was understood. Sewn on 3 alum toward supports that were then laced into parchment cover to connect A to B. With that cover removed, we can clearly see that the primary sewing is not on those alum-tooled supports. We have 6 cord double supports here. And then we can also see there's been some repairs made in situ in this blue thread around one shoulder and this white whipped thread around another shoulder. And we can see that that binding starts to look like a fairly quickly executed repair. It was in fact attached by 3 alum-tooled, we're calling them tackets, that have been driven through the shoulder of the text block at each side, probably about a quarter of the way through the text block, to attach that wrapper onto the text block. We can also see these leather patches that have been adhered over the supports, which made us think perhaps there's an earlier binding here. We've thought of a slotted structure, so which would see the sewing supports protrude through slots on a parchment binding. So that's one possibility. And when we looked at those leather patches under UV light, we could see what looks like a number 1 and a number 6 on this lower panel here. We wondered if perhaps this might have been an identifier or a fraction of a call number, perhaps when the manuscript was without a binding. In terms of treatment carried out, the sewing was stabilized in that central portion. Quires were repaired off book and reattached to extended supports, and then the manuscript was placed into a 3-part laced case paper binding. As I say, I don't have time to go into the details of conservation treatment in this presentation, but if you are interested, there's been an article published this year in the Bookbinder Journal, which goes into a bit more detail. So back to gutter debris. At the beginning of the conservation project, I'd just been working with Eileen and Kate on their project, which Eileen will explain in a moment, which was looking at examining pollen in manuscripts with a view to understanding usage. And I thought, well, maybe I can sneak into their project. So when I was doing just a light surface clean, I gathered the debris from each of the sections of the manuscript, so the historical chronicle and the pictorial history, in separate little baggies, and then asked Eileen if she would look at them to see what she could find. I will say that my sampling technique was less than optimal. I just brushed it into a collection tray and then brushed that into a bag, which now if I look back and I think about how lovely an airborne pollen is, then it's probably not the most optimal sampling technique. But thankfully, Eileen has a much better sampling technique, which I will now hand over to her, and she can explain to you her side of things. Thank you.
Okay. So as outlined in the introduction, I'm a lecturer in environmental geography. So I normally look at reconstructing past environments, and that's how I would use pollen. So this project with Kate was something quite really different for me as someone who normally uses pollen from peat bogs and lakes. I'd never even considered that they would actually survive in books. So when she asked me to take on this bit of her living, I said, well, why not? Why not give it a go? Let's just have a look and see. So very thankful then to the University of Glasgow Library and Keira then to help us kind of work out the method, the sampling method, because we needed a way that we could sample the books really nicely that wouldn't sort of destroy the manuscripts, because that really wasn't going to be helpful, but also that would be a means to kind of collect the relevant material. And lots of questions about whether the sampling method would work, would pollen even be preserved, all of those types of questions. And so that's when we used a couple of books from Glasgow, and they actually already sort of had flowers in them. So there's a little video to come next that kind of shows you the sampling technique.
You ready?
Yes, go.
We enlisted the help of Bob McLean at the University of Glasgow Library. He suggested a number of books that were likely to contain pollen so that we could begin testing the process on actual special collections items. This book, also from about 1900, which is a souvenir volume of flowers and views of the Holy Land, consisting of 12 chromolithographs depicting various places in the Holy Land, that has pressed flowers from each of these places mounted onto the facing plate. And the question now was whether that pollen could be extracted safely and whether the book had preserved the pollen grains sufficiently so that they could be identified. To ensure that our practices were safe for the rare books and to help develop the technique, we worked with Keira McKee. I had a moment where I thought, well, maybe we could put a protective screen or a mesh over the flower samples and vacuum lightly through that. But I think there's too much risk to the samples from abrasion and catching them if it were to slide or move. I think it's again quite a catching surface. So moved away from that. There's some little particles just around here that are lifting onto the brush. I don't know if that's just the. Oh, I guess a bit. Okay. Oh, PCL 14138. Okay, now let them move. Yeah. Are they the sorts of things you'd be looking at? Filter.
So I don't know, I don't know if you could hear it or not. The sampling is actually just using a little hoover because this is what I came up with. And as I say, I didn't really know what was going on in the kind of museum conservator world, but I understand that many of you already use hoovers. So in that sense, this is nothing new. But what I wanted was a mechanism that would allow the material to be retained on a filter. So I've adapted just a normal handheld vacuum cleaner, and then I got a little attachment made, and then a very simple filter that's borrowed from environmental sciences, and then some tubing, some flexible tubing, and then the filter. So if you could have the next one, next slide. And then you can see there's the filter, as I say, from water sampling, and then there was a Whatman paper filter then put in the filter paper, and that allowed me to trap the sample, retain the sample, and then also if I wanted to look at different sections of a book, different pages of a book, then we could use that. And then the technique of just using the brush to brush along the gutter and then run the hoover along behind it with the flexible tubing meant that I was pretty confident that I was collecting material that had sort of fallen in between the pages and had been retained within the gutters. And I think this helps in terms of the sampling, because it means that it's a bit more, it's more comfortable for those of you who manage these beautiful books and manuscripts, that I'm not interested in sort of running the hoover over the pages, that actually it's the gutter, the bits the material rolls and falls into that's actually the most relevant. So Keira had done the sampling slightly differently for the book, the material that she sent me. And then we've been out sampling using this method. And this method actually retains pollen really beautifully. And the two books that you saw being sampled in the video clip there, they did produce very nice bits of pollen, which is what we were expecting, because these actually had flowers, you know, on the pages themselves. But what that told me was that pollen does actually preserve, and that's the bit I had some concerns about. So could I have the next slide, please? So when I looked at the material that Keira had sent me then from these books, again, I had to think about what was to be a simple prep method to just make sure that I didn't lose any material. But I was also really aware that this material is, you know, it's part of the book, it's part of the whole kind of sort of object itself. And so I didn't want to destroy it. So any material that was too large, I sent that back. I was able to dry that and then send that back to Keira. So I was primarily interested in looking for pollen in these slides, because Keira had posed the question about this particular book, had it come from Mexico or not. So I was using what I would normally do in terms of the way I would look for pollen, and that's just simply using a light microscope. So that's just what I did. So I mounted the material onto a slide and I just started to look down the microscope at it. So very simple, nothing fancy at all. Can I have the next slide, please? So this is some of the images from the pictorial section. And I think what really surprised me was the amount of material that was in these gutters. So, I mean, I wasn't expecting anything quite so diverse. I mean, there's a real mixture of plant fragments and then spores and then very obviously modern materials as well, all found within this. And this is from the pictorial section. And Keira's question was around were there differences between the sections. So in the pictorial section, I didn't find as much material, but in the historical section, which is the next slide, I found much more debris. And if you'd looked at the bags that Keira sent me, then you can see that they were quite different in terms of just the amount. And they were also quite different in terms of the type of material I was finding. I found way more fungal spores, for example, lots and lots of human hair. And I did find one pollen grain. So there it is. You can see it. There it is. Little cedar pollen is actually. So at some point, this book was in an environment where pollen was going through the air, and it happened to be cedar, which is a Mediterranean pine species. So this book has been to the Mediterranean. But I think what impressed me more was just the range of materials. And I got quite excited because I thought maybe these little round, clear cylindrical things were parasite eggs. So this ties to our previous speaker thinking about, well, what you get that tells you about the, you know, the people. And I thought maybe it was yeasts. And then Keira said, oh no, she thought it was just starch grains, which I understand are used in book kind of conservation. So that was a bit disappointing. But I think just looking at this material, gives you a slightly different perspective on the potential of the dirt, the things that are part and parcel of the way in which these books are being used. And then what does that allow you to say about the books and their use and their life and the things that they've been through. So the fungal spores, for example, would be quite interesting to start thinking about. Can I have the next slide? So, so we have this method then that's actually a quite easy, straightforward way to sample these particular manuscripts, and then thinking about collecting the material from specific parts of a book or sections of a book. And if pollen is what you're interested in, then this will work best if you think the books have been outside, or if you think the books have traveled. So I'm currently looking at another book, and this is part of Kate's Leverhulme, to see if this book has traveled. And this book also contains pollen, but it's not evenly throughout the book. It's in specific sections. So again, that points to using this technique to say something a little bit about what you think has happened to this book. We also got a little bit more money just to go and sample some more books. So this was a little bit of money that came from NERC, and that was just to look again, just to get out and just sample some more books. And so I'm looking at those samples just now as well. I think the thing to stress here is that they're not proof, they're just clues, they're just extra bits of evidence, little pieces of information. And it's the diversity of the material, I think, is what astonished me most. And then, thinking about the dirt that maybe is quite often cleaned and then disposed of, maybe this project will change your attitude to the dirt and dust that's in these gutters, and what can you actually use it for, and is it useful? It might not even be useful at all. I might just find this really interesting and nobody else does, but I think the pollen's working better than I expected. We still have all sorts of issues along the way, but I think curiosity can see you a long way.
Okay, this is Keira again. I'm just going to hop in and say about next steps in terms of the of Hunter 242. So hopefully at some point we'll be able to look at the rest of the sample from Hunter 242. I think, Eileen, if I'm right in saying that you looked at 1 or 2 slides from each section of the book,
yeah.
So we still have some of the sample to look at. Maybe we'll find some more pollen. Maybe we won't, but it'll still be interesting to look. We'll be making the results of that available for researchers who are interested in that manuscript. During the conservation project, we had a special interest group formed of academics and art historians, people who had an interest in that manuscript from different disciplines. We also are hoping to analyse some textile fibres that were removed during conservation in terms of fibre analysis and dye analysis, especially from that pictorial section of the manuscript, to see if that gives us any clues about the materials that may have been used in binding, and if that can point to a location as well. And then we'll be adopting Eileen's lovely improved workflow for gathering dust debris, which will replace my my fluffy goat hair brush, and hopefully just procure better samples to store alongside books. And we're hoping to try doing that adaptation with the tubing with museum vac, which are the vacuums that are more commonly found in conservation studios. So if we can crack that, we'll certainly share that as a how-to. So Eileen, do you want to talk about the next?
So yeah, so the next steps then for the projects that just finished, the materials that I've collected from the books that were in the Netherlands mainly, they are going to be used as part of an exhibition for to illustrate the life of and use of the book. So again, this might be just an interesting way to show the evidence, to show the material. It may not, aid in terms of the way you view, but it might be just something interesting about, the way this material collects, and sort of the whole kind of forensic thinking about, well, what does it mean? Can you say something a little bit extra about these particular books? And then another colleague from University of Stirling, Jenny Morgan, she works in heritage, and she is interested in attitudes to dirt, on objects, on books, and maybe by thinking about this slightly differently, our attitudes to cleaning and to dirt, in particular on heritage objects, changes slightly. So that's her side of this project.
Okay, that's us. So we'd be happy to answer any questions. I'll stop sharing my screen. Okay.
Thank you both very much. That was really, really interesting, and it just, as the other papers today have shown, just emphasizes the importance of that conjunction between the knowledge and expertise of conservators, of manuscript scholars, of with John's paper, for sort of photographers and imaging scientists, people from outside of the bibliographical and manuscripts world, like yourself, Eileen. It's really fascinating. Does anybody have any questions? Elaine Wright raised her hand. If you'd like to put your question to Keira and Eileen, Elaine, please feel free to use the Q&A function. I thought that point that you made at the end about the kind of questioning the problematic, our assumptions that dirt and dust and grime in books is problematic, I thought was an interesting one to conclude with. I mean, I suppose, you know, the conventional wisdom is that we should all move to having our manuscripts in boxes, and they should be sort of hermetically sealed, and, you know, so that the our own day-to-day grime isn't getting in there. But I suppose we'd unintentionally be stopping future researchers from understanding how we're using the materials if we're trying to make as little impact on them as possible. It's like there's a kind of tension there, isn't there? We know something about how they're being used because of they've not been used in the kind of hermetically sealed environments. No. Okay, we've got some questions. John Hodgson wants to know, is there any way to date the pollen samples and thus create a kind of chronology of the manuscript's travels?
Yes and no is the answer to that one. So no, in that it's very difficult to directly date the pollen itself, but yes, in that if you get a plant assemblage that tells you something about plants that were grown nearby, and maybe they're not grown anymore, and you might have historical records of when they were growing, then you could say that that at that point in time the pollen arrived at earlier, and then then as the plants shift and change, then you have a later pollen assemblage. So you can do it that way, but that relies on lots and lots of lovely pollen grains arriving on your book, and so the answer is a tricky yes and no to that one, I'm afraid.
I think that might answer another question that's been put up by Rowan Watson, which is, you know, is 16th century pollen of one kind identical to 20th century pollen from the same plants? Well,
yes, it will be, but this depends on how well the pollen is preserved. So new pollen looks very different to pollen that's been there for, you know, several years, if you see what I mean, because the pollen that's been there for years is all degraded, and new pollen looks very different. But that would be really very recent pollen, so last year or so compared to older pollen. So you get a sense of how long the pollen has been there sort of relatively, but no, you wouldn't know exactly what year it arrived in. But for my discipline, actually, you know when the book was made normally, and I never get that close, you know. So I'm really excited that this pollen has arrived within the last, you know, 200, maybe 300 years, you know, so that's good enough for me. But I understand that maybe for the use of a book and that over time, it's maybe you want to know a little bit more. So that's why this sits alongside other evidence that you might have for the way in which these books have been used and their change in use. So the book may have traveled from one part of the world to another, and then the pollen should show that it's different from one part of the world to another part of the world. If it's traveled in and around Europe, that's more difficult. But if it's been to Southern Europe, then that's very different plant communities than in Northern Europe, for example.
Great. Yeah, I think, you know, the work that Matthew and Sarah have done, particularly at the Borthwick, has been where it's all documents which are generally all dated. You know, they've benefited from precisely that same conjunction of the kind of the material evidence and the kind of documentary evidence, providing that useful dating criteria, which in other circumstances you just don't get.
No, you've got different questions, so yeah.
So Glenn Bartley has asked, how do you see institutions collecting and archiving their own samples? So, you know, I suppose, do you have some kind of advice about what?
Shall I take this one, Eileen? Yeah. So from a library point of view, if that's the question, so we've already been gathering our gutter debris here for a period of time. I see that someone else has also said they feel guilty about throwing it away. I've certainly done that in the past as well. But over the last few years, we've been gathering it and storing it in little polyethylene bags or sealed in polyester, just like edge welded, and then either storing that alongside the book, or we have an extra repository where we're storing them kind of and giving the call numbers of the book, and they're in their own store, like gutter debris that's associated with particular manuscripts. I think what we're suggesting, and there are different institutions that do that in different ways, but I think what we're suggesting here is really an improved methodology for collecting that sample. And there will be different ways to how those samples are stored. But I think for myself, what I've done is the brush into a catching tray. I'm sure there are other methods. And then it's kind of brushing that into a bag. And I think the amount of, especially the smaller material and lighter material that is probably lost during that collection process is probably not good. And so what we're suggesting, like if we adopt this method of hoovering, maybe before subsequent surface cleaning, then it gathers that material in a more thorough way that can then be sealed and stored according to the institution's own practices.
Thanks very much. We're coming up to time, so I'll leave the remaining questions in the chat, and Keira and Eileen, I think you should be able to kind of type your answers or send details there if you like, but the questioners are also welcome to contact the speakers directly. Thank you both very much. It's a really interesting paper.
Thank you.
So now I'm going to have to multitask. I not only have to introduce myself, share my slides, and get my script up. So if you will bear with me for one moment. So, okay, hopefully everybody can see my slides. Today's final paper is given by me. So I'm James Freeman. I'm the medieval manuscript specialist at Cambridge University Library, and a chair of AMARC. And I'll be presenting on scientific aids for the study of manuscripts, R.B. Haselden and the history of heritage science. The name of Reginald Bertie Haselden is now not widely known outside of the Huntington Library in San Marino, California, where he worked as curator of manuscripts from 1923 until his death nearly 30 years later. Before he emigrated to the United States, he'd served in the British Army during the Great War and brought some of that military spirit to his library work. Reggie to his acquaintances, but Captain Haselden to his colleagues, he expected his mostly female staff to stand to attention by their desks for inspection each morning. The atmosphere at the Huntington is much more relaxed nowadays. Haselden was also the author of Scientific Aids for the Study of Manuscripts, which he published in 1935. It's a guide to heritage science, long before it was described as such or entered the mainstream of manuscript studies. In recent years, Haselden wrote, scientific knowledge has extended its sphere of usefulness to almost all fields of endeavour. The question is whether this knowledge can be utilised and brought to bear on the complex problems encountered by the paleographer and the student of literary and historical manuscripts. Scientific aids describe the setup and use of different lighting and colour filters, microscopes, ultraviolet light, photography and measuring instruments in the service of research, identifying forgeries, or diagnosing damage, mould, or pest activity. As a foundation for this specialised work, Haselden also introduced the study of manuscripts through its various, as he put it, auxiliary sciences: epigraphy, paleography, philology, chronology, diplomatic, heraldry, and something he called bibliotics, which we know now as codicology. He also set out some principles and practices for the proper treatment, handling, storage, and repair of such materials, what would nowadays fall within the remit of conservation and collection care. Introducing his and his colleagues' experiments, he was cautious to advise that particular instruments will be described solely for the purpose of illustrating technique and are not specifically recommended by the writer. Nevertheless, scientific aids exudes a machine-age confidence in the applicability of science, its equipment and its methodologies to any area of inquiry. A dizzying parade of custom-built and proprietary apparatus marches through its pages. Lights lamps, Spencer lamps, an ultra-opaque illuminant system, Hanovia ultraviolet lamps, Lovibond tintometer glasses, a Zeiss aplanatic non-spherical reading glass. I wonder if there's a room in the Huntington's basement where these once treasured products are now gathering dust. And Haselden hoped that his examples would stimulate other and more competent investigators to experiment still further with scientific instruments in the field of manuscript study. Arising from a very particular set of circumstances, scientific aids for the study of manuscripts affords valuable insights into some of the problems and priorities in the Huntington Library's early years. However obsolete the equipment, the development of those tools and techniques is also part of the broader history of heritage science. That is, the ways in which technology, in whatever form, has been used to expand our knowledge of manuscripts' origins, production, contents, provenance, or use. As such, scientific aids offers us an opportunity to reflect upon our own practices as curators, archivists, and conservators. What principles guide the use of collections within our care, how we balance the competing demands of research, reproduction, and conservation, and as scientific approaches proliferate and become increasingly accessible, how we facilitate but also regulate and record their use. But what of Haselden himself? Reginald Bertie Haselden was born on the 21st of May, 1881 in Beckenham in Kent. He was educated at the Abbey School there before going up to Cambridge in 1899. He left Trinity Hall in Easter 1902 without taking a degree and moved to London, taking employment in the city as an authorised clerk to stock jobbers. Then in his mid-20s, Haselden fell in with Aleister Crowley, of all people, a cultist and experimental practitioner of sexual magic. Haselden's recorded in August 1909 as the 9th member of Crowley's mystical Kabbalistic organisation, known variously as the Order of the Silver Star, the Argentium Astrum, or simply the AA. Haselden's interest in the order isn't wholly clear, but he remained a member until at least October 1912, earning promotion from probationer to neophyte, meaning, apparently, that he had acquired perfect control of the astral plane. Crowley's interest in Haselden, meanwhile, is perhaps more obvious. His 1910 collection of verse, The Winged Beetle, contains a poem dedicated to Haselden that begins, "How should I seek to make a song for thee when all my music is to moan thy name?" The connections between Hermetic writings, magic, and the Middle East that inspired Crowley and his acolytes might explain Haselden's collection of Egyptian antiquities. These included an ostracon depicting the Egyptian crocodile god Sobek, which you can see in the bottom left-hand corner there, several ushabtis, these are funerary figurines intended to serve as minions for the deceased in the afterlife, a mummified falcon, and, strangest and most morbid of all, a mummified hand. Medieval manuscripts and early printed books were another early interest. Don't worry, we've dealt with the occult, we've dealt with the Egyptian antiquities, now we're on the more familiar territory. So either life as an authorized clerk was well remunerated, or Haselden came into some inheritance, but by the mid-1910s, he was in business as a dealer, operating out of premises on Sicilian Avenue and later Earl's Court Road under the pseudonym H.A. Selden. The Post Office Directory for 1913 has him down as a second-hand bookseller, which I suppose is true in a sense. Medieval manuscripts and incunabula are about as second-hand books come. Several items to pass through his hands ended up at Cambridge University Library, though not always directly. A couple of these came via Sir Stephen Gazeley. These included a fragment of an 8th century copy of Isidore's Etymologies, a fragment of a 9th century Latin glossary, and a 1497 printing of Henricus de Hassia's Secreta Sacerdotum, the only copy of that edition now to be found in a UK library. My greatest finds have been in furniture, rag and bone, and booksellers' shops, he told the university librarian Francis Jenkinson. I always use a small bottle of chloroform to kill the bugs and fleas and other pests which attack one in these places. For a brief period after the war, Haselden also worked as a cataloguer for Sotheby's. The sale of many aristocratic and private libraries was then providing plentiful and profitable business, and Haselden shortly followed the same transmigratory path across the Atlantic as many of those manuscripts and books, which journey projects such as the Schoenberg database, mapping manuscript migrations, and more recently Laura Cleaver's Cultivate MSS projects are illustrating. Following a short stint with Gabriel Wells in New York, Haselden was recruited in 1923 to work at the recently founded Huntington Library as curator of manuscripts. When he arrived in San Marino, the Mansion House, museum, and library buildings would have still been visible to passengers on a nearby line of the Pacific Electric Railway, whose distinctive red cars are commemorated in a nearby mural on Huntington Drive. The railway was owned by none other than Henry Edwards Huntington, eponymous founder of the library, museum, and gardens, on land that he had purchased as Rancho San Marino 20 years earlier. And you can see an aerial view of that area of Pasadena, or South Pasadena, in 1924. It sort of goes without saying that it doesn't look like that anymore, sadly. Railways and riches were in the family. His uncle Collis P. Huntington, who you can see on the right here, described by one contemporary as scrupulously dishonest, was one of the so-called Big Four, the financial backers of the Central Pacific Railroad that linked the East and West coasts in 1869. Collis died in 1900, leaving a third of his fortune to his wife Arabella, making her reputedly the richest woman in America, and a third to his nephew, Henry. Henry and Arabella shared a passion for European and especially English art and culture. To the shock and scandal of polite society, the two married in 1913, and they spent their remaining years in a frenetic spending spree, amassing a collection of over 11 million manuscript items, dispensing vast quantities of money, and often purchasing entire collections en bloc, like the Bridgewater Library, which I think Huntington paid $1 million for. The Huntington Library itself was founded in 1919 in order to accommodate these rapidly expanding collections, and it remained a private institution until after the death of Arabella Huntington in 1924 and her husband in 1927, finally opening to the public in the following year. Haselden may have seemed to Arabella and Henry to be another choice old world acquisition, a man apparently in possession of the right education, cultural hinterland, and commercial experience to take charge of the library's burgeoning collections. The details of that day-to-day work are preserved within the Huntington Institutional Archive, but scientific aids points to the library's priorities and some of its problems in those early years, and the motives behind the pioneering experiments that were conducted there. The Huntington's collections had been formed at speed and in bulk. These needed to be sorted, described, properly housed, and the library's pivot from a private to a public institution made this especially urgent. Furthermore, in early 20th century San Marino, just as in early 21st century Washington, DC, rapid and voluminous collecting did not always go hand in hand with due diligence. Fakes and forgeries made their way into the collection, and the opening of them to research made the identification of the rough among the diamonds an urgent necessity for the sake of scholarship and the institution itself. Scientific aids played a role in building that reputation, or building the Huntington's reputation, I should say. It advertised to the world that the new library, less than 20 years old, but already one of the most important repositories of manuscripts in the world, not only knew how to take care of its collections, but was leading the way in the most technologically sophisticated analysis of them. But scientific aids also reflected back the experience that had been gained over the previous decade, establishing and documenting procedures for the current and future benefit of the library's staff. If much of scientific aids reads like a staff manual, with Haselden's distinctive managerial tone jumping off its pages, that's because that is exactly what it was, at least in part. To ensure his staff were up to speed, Haselden provided an introduction to the field of manuscript study, and as he put it, a suggested procedure for the examination of a manuscript, which reads very much like a template for cataloguing. There was advice for good practice in the handling and care of manuscripts, and step-by-step instructions in how the scientific aids should be set up, fine-tuned, and used on the collections with appropriate care. And you can see here Norma Cuthbert, who was the head cataloguer of manuscripts at work in 1938. She donated a typescript that Haselden had prepared, which gave some of the basic information, introductory information about manuscripts and their histories. And it gives you a sense of what he was like to work for, that this was entitled "A Miscellany for Morons." The replicability of the instruments described in scientific aids was to some degree beside the point. Could other libraries have afforded to purchase and build the instruments that Haselden described, or dedicate the time to minute analysis of their collections? At that early and experimental stage, much of the equipment was inevitably custom built and designed. But without technical drawings or illustrative photographs, one wonders whether other institutions could realistically have reconstructed these setups. But there is still a value to recording such specific, even experimental work. We may trace the history of heritage science through Haselden's case studies or through the acknowledgement of his and his colleagues' work in the footnotes of scholarly publications that were built on findings that they had helped to secure. But by its nature, heritage science advances through testing and experimentation, the results of which are not always known at the outset and which ultimately may not prove successful. The success-orientated model of publication in the humanities does not comfortably accommodate such descriptive work, nor experiments that yielded no results. Yet that information can guide future work and experimentation. What we do in libraries and in our photography and conservation studios should be recorded in similar detail so that future curators, imaging specialists, and conservators can understand and build on our own work. Haselden recorded those processes by describing the components and construction of his equipment, and then step-by-step instructions for its use and adjustment. So in his chapter on microscopes and magnifiers, for instance, he explained how the ultra-opaque microscope could be used in the examination of paper, pen strokes, or pencil marks by using reflected light. It had to be placed over the object, focused carefully, and a suitable light filter added to regulate the intensity of the light. And he advised that casting the light at different angles could produce better results, which was something that John's paper earlier on reminded me of. These records also have a practical value for the conservation of collections. Thanks to scientific aids, we know that the Huntington Library disinfected its new acquisitions by placing them in a vacuum chamber, which you can see here, and fumigating them with carboxide, a mixture of ethylene oxide and carbon dioxide. And thymol vapor in a vacuum chamber was also used for the elimination of mold. Would we do that nowadays? What was its effect on the different collections? Stains were also sometimes removed Haselden tells us, by placing hydrogen peroxide in a concentrated solution on a flat block of plaster of Paris. The item to be treated was then held face downwards about 1/8 of an inch from the block, which sounds rather perilous to me. In most cases, this will bleach the stain, Haselden advised, although it may take a considerable time. This process, however, should only be resorted to when absolutely necessary, as it's likely to have an adverse effect on some types of paper and may bleach or turn into faint yellow, those writing inks which have an iron salt basis. Is there evidence where this has happened? Knowing how our collections have been treated or experimented on in the past is vital information that can inform and guide our treatments today. We fail to record these approaches at our peril. Scientific aids for the study of manuscripts reminds us that heritage science has a history, perhaps a longer history than we might think. The techniques that John, Keira and Eileen and Matthew and Sarah have described are at the very forefront of the heritage science field. They've each described highly sophisticated and highly innovative and creative technical, chemical or biomolecular analyses. Similarly, the 17 case studies in the final chapter of Scientific Aids demonstrated the results that Haselden and his colleagues had achieved, such as using UV light to correct a reading in the manuscripts of the Townley mystery plays, explaining changes in the colour of ink in a collection of Middle English poetry, or revealing deleted writing in a manuscript in the Hastings papers. As such, the history of heritage science is part of the history of manuscript studies. In very practical ways, it illuminates the questions that were being asked of manuscripts at a particular time and in a particular place. And just like the history of paleography or codicology, it enables us to understand how our knowledge of manuscripts has been developed and advanced. The scientific instruments that Haselden described might seem crude or clumsy compared to what we have at our disposal a century later. And I can't help but wonder what we'll have at our disposal a century from now, seeing what we've seen this morning. But the use of these scientific instruments was motivated by the same objective. In Haselden's words, the solution of problems of interpretation relating to the text, physical history, and provenance of manuscripts. Conceiving heritage science in terms of its objectives rather than its methods enables a more expansive and more informative history, the one that concentrates on the non-invasive, non-destructive techniques that we now use. For the purpose of revealing hidden or faded text, for example, the history of heritage science encompasses everything ranging from modern-day multispectral imaging, the illuminants and light filters, ultraviolet light and magnifiers that Haselden documented, but also reagents, whose traces we can still see in manuscripts today. My point is that the history of heritage science is also part of the history of heritage culture and reveals how our attitudes to the manuscript page have changed and evolved. Scientific Aids was published at a transitional moment in the formulation and application of non-invasive and non-destructive principles that guide our work today. Haselden considered reagents objectionable, quote, because their effect is always uncertain and they sometimes even damage the manuscript. But nevertheless, he suggested ammonium hydrogen sulfide might be used as a possible last resort. He even recorded his own experiments with two chemicals, anthracene and esculin, which you can see on the left here, that he had been recommended for use by a colleague at Caltech. The latter caused the stronger fluorescence, he found, but its precise effect on the paper or vellum has not yet been ascertained. Therefore, its use cannot be recommended until further experiments have been made. We would do well to emulate his cautiousness. We cannot know in our own short-term lives the long-term effects upon the manuscripts of the work and the experiments that we conduct today. This represents a risk not only to the manuscripts, but the broader scholarly field, a risk that Haselden perceived. No examination can be justified which in any way injures the manuscript, he wrote, rather contradicting his recommendation of the use of ammonium hydrogen sulfide. But anyway, if the manuscripts cease to exist or cannot be traced after they've been quoted, their evidential value is considerably minimized and may be entirely discredited by later scholars. Placing multispectral imaging on the same continuum as the dreaded reagent reminds us that our attitudes to the manuscript page are continually developing as our understanding of them and our own actions develops too. Witnessing the work of Haselden and his colleagues through the window of scientific aids reminds us to reflect on the acceptable compromises we still make in the handling, in the use, in the digitization, in the scientific analysis, and in the study of materials that we know will fade, that we know will degrade, and that we know will eventually disintegrate. Thanks very much. That is the first time that I've ever read a conference paper off my phone, but all the printers at the UL were not working yesterday, so I couldn't print it out. So, okay, I'm happy to take questions if anybody has any. I'll just give people a moment. If anybody ever has the chance to visit the Huntington Library, I can't recommend it highly enough. It's an absolutely incredible place. Yeah, I had the privilege of going there to look at one of their manuscripts a few years ago, and the identification of that manuscript as the author's own working copy was as a consequence of the kinds of techniques that Haselden had pioneered. So it had a kind of material bearing on my own work. James Morrie has asked about where I sourced the photos. So Jim, they're all on the Huntington Library's digital collections platform, and some are also from the Calisphere City of Los Angeles digital platform as well. And they have specific subsets that address the Huntington Library's history. So yeah, that's where they're all from. So Rosamund McKittrick's asked if Haselden discussed the disastrous efforts with reagents made in 18th and 19th century by Angelo Mai. No, he didn't. No, he doesn't give the sort of historical perspective in that regard. But I'm aware of those experiments myself, and I've been picking up other instances of their use. I'm grateful to Andrew Prescott for pointing me in the direction of information about their use at the British Museum and later the British Library, and also at the Public Record Office as it then was. So I think, yeah, an interesting history of reagents could be written. Interesting, if slightly horrifying. There you go. Oh my goodness, Barbara Bienius has asked me, what do I perceive as the biggest challenge with curating manuscripts today? Oh gosh, how long have you got? We're going to have to stop in 4 minutes, Barbara. I don't know how to answer that. Yes, I think one of the challenges, but also one of the kind of most promising and exciting things, is that working as a curator, you're actually you're the kind of node point between all of these different areas of expertise, and everybody has got their own slightly different approach to manuscripts, whether they are imaging scientists and photographers, whether they are conservators, whether they are heritage scientists, whether they're also sort of software developers and computer technicians in that way. I mean, to say nothing of the actual researchers who do the research on the stuff itself, you know, in the kind of traditional sense. And yes, you're in the middle of all of those different competing interests and competing demands, and keeping pace with what's going on with the collections, I think is probably the biggest challenge. And I can't pretend I meet it. A question from Suzanne. This was the golden age of Hollywood. Is there any connection with Haselden's innovative cameras and imaging experiments? Not that I know of, but the Huntington seems to have been particularly well situated to kind of take advantage of those technological innovations. So Haselden refers in a number of places to collaborations with colleagues at Caltech, with technicians based in nearby Pasadena who had developed, or tweaked particular bits of proprietary equipment. And they also employed internally expert photographers as well. And it's kind of interesting, actually. Haselden isn't very much known, but his colleague who you saw in my slides, Ludovic Bendixson, is. And studies have been written on Bendixson's photographic work. So yeah, so there's in a way there's a kind of recipe there for our own success in the present, in integrating heritage scientific or photographic or conservation expertise within our own libraries, as well as collaborating with expertise beyond the walls of our own institutions. Okay, we're coming up to 1:30, which I think is the end of the time that we've got. Thank you everybody for the questions that you've put to me, but also thank you to the audience for coming and for the questions that they've put to the speakers. And thank you very much, of course, to the speakers for taking the time to share their work. I think it's been a really interesting and stimulating meeting. And you're all very welcome to come to AMARC meetings in the future if we have them in person or online. We hope to see you there, and hope you all have a very nice weekend. And thank you again. And thank you also to the Bodleian for their support in hosting and organising it.